Hi, everyone. I'm going to talk about chapter 6, the second part of the cardiovascular system, specifically the blood. Sometimes we call it the circulatory system, but it is still part of the overarching cardiovascular system. So an overview, of course, is the first section. So the heart pumps 75 milliliters of blood with each contraction. On average, the heart beats about 70 times per minute. Therefore, the heart pumps roughly 5,250 milliliters per minute. If you want something to compare that to, I put the Coke bottles on the right. You know what a 2 liter looks like. So 2 liter is 2,000 milliliters. So that representation, about 2.5, a little bit more than 2.5 bottles of Coke, the big 2 liter bottles. That's how much our heart pumps every single minute. So the entire blood supply is circulated each minute. Functions of blood, you got transport, the oxygen, nutrients, wastes, carbon dioxide, hormones, defense. We defend ourselves against invasion by pathogens. Some white blood cells phagocytize microbes, which just means they eat them. Others produce antibodies, which are proteins that will disable pathogens. Our blood protects against fluid loss by clotting. And then regulation, ever-present homeostasis. Temperature, blood pH, water balance, our blood plays a part in all of that. Blood is a liquid connective tissue made of formed elements, the cells and the cell fragments suspended in plasma. The formed elements are produced in the red bone marrow. You got red blood cells, white blood cells, platelets, and their fancy science names. We'll talk about them later. There's also some stem cells floating around in there. Plasma consists of 91% water and then 9% salts and organic molecules. Solutes help maintain the osmotic pressure of blood. Solutes like salt. Salt will act as a buffer to bring the pH back up or back down, depending on if it's too high or too low. Other solutes like nutrients, wastes, and hormones are also present. Plasma proteins are the most abundant organic molecule. Most are produced by the liver, and they create osmotic pressure in the blood. There are 3 major types of plasma proteins. Albumins are the most abundant. They contribute to osmotic pressure more than the others, and they transport molecules in the blood. Globulins, some transport substances in the blood. Others, like gamma globulins, fight pathogens. And fibrinogen is inactive, but when it is activated, it will form blood clots. The next section is all about the RBCs, the red blood cells. So erythrocytes is the fancy science name for red blood cells. It has a biconcave shape, which increases surface area. It's an unusual cell type in the body due to hemoglobin and a very unique internal structure, and it is very abundant. We have millions upon billions of red blood cells in our bodies. Red blood cells are specialized for oxygen transport. It contains the protein hemoglobin, which is a pigment that binds oxygen. And then the word pigment signifies color. So hemoglobin is the pigment that is responsible for the red color of blood. The heme portion of the hemoglobin molecule will bind up to 4 oxygen molecules. That's what the little GIF in the bottom corner is representing. It will also bind carbon monoxide. When bound to oxygen, hemoglobin is called oxyhemoglobin. When oxygen leaves, it is called deoxyhemoglobin. And when it is bound with the carbon monoxide, it is called something else, which we will talk about. So there's our red blood cells, very close-up structure. There they are traveling through capillaries one at a time. And then that's kind of a little translucent X-ray of a, not really an X-ray, but you know what I mean, of a red blood cell that's what the hemoglobin looks like, with the 4 little iron molecules in there, and each iron molecule will bind an oxygen, and that iron with the oxygen is called a heme group. So 7% of carbon dioxide is transported in dissolved plasma. 23% binds to the globin portion of the hemoglobin. Hemoglobin bound to the carbon dioxide is called carbaminohemoglobin. 70% is transported in plasma as bicarbonate ion, the HCO3-, and that bicarbonate ion is what acts as the buffer for the blood pH to maintain our blood pH at homeostasis. Red blood cell production occurs in red bone marrow. As RBCs are produced, they lose their nucleus and most organelles. Without a nucleus, it can't make proteins for cell repair, so they only live about 120 days. So we are constantly having to replace our red blood cells. Old worn-out cells are removed from circulation by macrophages in the liver and spleen. The disc shape allows them to squeeze through small capillaries and allows for maximum surface area for gas diffusion. You can see the little gif of the red blood cells squeezing through that small part. Erythropoietin, or EPO for short, is a hormone that is produced by the kidneys when oxygen levels of the blood are low. It will stimulate the bone marrow to produce more red blood cells. So this is one way that our blood works with our kidneys, our urinary system, to maintain that homeostasis. Disorders involving red blood cells. There's jaundice, an accumulation of heme in the blood if the liver can't excrete it. Our skin and our whites of our eyes will turn yellowish. It's very common in newborns. Anemia, too few RBCs or too little hemoglobin. Iron deficiency anemia is the most common form. It just from an inadequate intake of dietary iron, so hemoglobin can't be made. Pernicious anemia is a lack of vitamin B12, which is needed to make red blood cells. And folic acid deficiency anemia, we need folic acid to make red blood cells. If we don't have enough, our red blood cells suffer, our red blood cell count. Hemolytic anemia is too much hemolysis or rupturing of the red blood cells for some reason or another. And then sickle cell disease is a genetic disease caused by which causes red blood cells to become sickle-shaped. The sickle cells stick together and cause blood clots. They're also prone to rupture, and it is due to a mutated hemoglobin structure. It is an actual mutation. The next section is about white blood cells, WBCs. So its fancy science name is leukocyte. They are large cells. They have a nucleus. They are translucent unless we actually stain them, which of course we do when we want to look at them. They're less numerous than red blood cells, but just as important. They're produced in red bone marrow. Production is regulated by colony stimulating factor. They fight infection and are an important part of the immune system. Some live only for days, while others live months or even years. There's two categories of white blood cells. There's granular leukocytes, which contain noticeable granules in them, or grains. And then agranular, they don't have the grains in them. We will go through all 5 of these. And grains are just the tiny little dots in there. Neutrophils is the most abundant white blood cell. About 50 to 70% of all white blood cells are neutrophils. They have a multi-lobe nucleus. They are the first responders to bacterial infections. They engulf pathogens by phagocytosis, and they can leave the bloodstream, so sometimes they're found in the interstitial fluid and the lymph. So on the left is kind of an animated picture of it, and on the right, if you actually look at cells, at blood under the microscope, and you find cells that look like this with the multi-lobed nucleus, those are neutrophils. Eosinophils have a bilobe nucleus. They have many large granules. Its function is parasitic infections. They also play a role in allergies, but not the biggest role. So again, on the left, there's your cartoon version. On the right, that's what it looks like under an actual microscope. Has 2 lobes and grains. Basophils are the rarest of white blood cells. They have a U-shape or lobe nucleus. In connective tissues, basophils and mast cells release histamine during allergic reactions. And histamine will dilate blood vessels, but constrict breathing passageways, like during an asthma attack. On the left is your cartoon version. It has the biggest grains or granules of all, and sometimes the nucleus is kind of obscured because those grains are so big under the microscope. Lymphocytes. About 25 to 35% of all white blood cells are lymphocytes. There's 2 types of lymphocytes, B cells and T cells. B cells, when mature, produce antibodies, which mark a pathogen for destruction. And some T cells directly destroy pathogens. We'll get into those when we talk about we will get into those in more detail when we talk about the immune system. But under a microscope, it just looks like it has one huge nucleus, and there's no grains. And then monocytes are the largest of the white blood cells. In tissues, they developed into even larger macrophages, which engulf pathogens, old cells, and debris. And the monocytes, they can be mistaken for maybe the eosinophils with the bilobe nucleus, but they look more like a horseshoe. It's a little bit thicker, and there are no grains. Disorders involving white blood cells. SCID, or severe combined immunodeficiency disease, is an inherited disease in which lymphocytes just don't develop at all. This is more commonly known as the bubble boy disease, where he lived in a bubble, in a plastic bubble. This is treated with stem cell therapy nowadays, but in the 80s, there was no treatment. He had to live in a bubble. Leukemia is a group of cancers in which white blood cells proliferate without control. Most of these white blood cells are abnormal, so they don't function well. You would think we have a lot of these white blood cells, so we would be really healthy because we would never get attacked by pathogens, but that's not how it works. The white blood cells don't really work properly. Infectious mononucleosis, the Epstein-Barr virus, infects lymphocytes, results in fatigue, sore throat, and swollen lymph nodes. Next are the platelets, whose fancy science name are the thrombocytes. These result from fragmentation of large cells called megakaryocytes in the red bone marrow. They're not actually true cells, but they are a fragment in our blood, and they do a lot of stuff for us. There's about 2 billion platelets that are made per day, and its function is blood clotting or coagulation. There are plasma proteins, prothrombin and fibrinogen are also involved in blood clotting. It is a cascade of events controlled by positive feedback mechanism. It is important so that the plasma-informed elements don't leak out of broken blood vessels. There are 13 different clotting factors, calcium ions, and enzymes that participate in clot formation. When a vessel breaks, platelets will clump to partially seal it. Platelets in injured tissues release a clotting factor called prothrombin activator, which converts prothrombin to thrombin, and this requires calcium. Thrombin acts as an enzyme that converts the plasma protein fibrinogen to fibrin. The fibrin thread provides a framework for the clot. Red blood cells get trapped in this framework. The fibrin clot is temporary, and an enzyme called plasmin will destroy the fibrin network so that tissue cells can grow to repair the vessel. Then serum will escape from the clot, and this contains all the components of plasma except the fibrinogen and prothrombin. And again, a picture is worth 1,000 words. I'm going to play this couple-minute video to explain the coagulation cascade.
Hemostasis is the normal physiological response to limit blood loss after vascular injury. Blood clots are made up of activated platelets, red blood cells, and fibrin, which hold the clot together. The process is mediated by around 30 proteins, which are known as clotting factors. Platelet activation and aggregation begins when vascular injury exposes collagen and von Willebrand factor from within the vessel wall to the blood. Platelets adhere to these proteins and become partially activated, changing shape and releasing signaling molecules which recruit more platelets to the site. At the same time, a protein called tissue factor triggers initiation of the coagulation cascade, the process that leads to full activation of platelets and the generation of fibrin and clot formation. Vascular injury causes tissue factor on the outside of cells in the vessel wall to come into contact with factor 7A and factor 10A in the blood, leading to the production of a small amount of thrombin. This thrombin fully activates platelets and triggers a cascade of clotting factors that activate each other, culminating in factor 10A-mediated generation of a larger burst of thrombin production on the platelet surface. One molecule of factor 10A leads to the formation of 1,000 molecules of thrombin. Thrombin converts the soluble protein fibrinogen into insoluble fibrin strands at the site of vessel injury, while at the same time activating factor 13 protein to cross-link the fibers. The fibrin mesh stabilizes the clot and traps more platelets and red blood cells, facilitating its growth. In normal physiological conditions, The process of clot breakdown or fibrinolysis begins as the structural integrity of the vessel wall is restored. Endothelial cells begin to secrete a protein called tissue plasminogen activator, which activates the protease plasmin to degrade fibrin, dismantling the structure of the clot and restoring the integrity of the vessel.
So, as I said, a picture. I like animations. I'm a visual person. I'm hoping some of you are too, and those animations help a little bit, help explain some of these concepts. But disorders related to blood clotting, thrombocytopenia, are too few platelets due to not enough being made in the bone marrow or the increased breakdown outside of the marrow for some reason or another. It can be caused by leukemia or drugs. The symptoms is just excess bleeding. The clot is just not forming for some reason. A thromboembolism, when a thrombus, the stationary clot, forms. When it travels, it's called an embolism and then plugs another vessel. Hemophilia, there's version A and version B. Christmas disease, von Willebrand disease. These are genetic deficiencies of a clotting factor. Like you heard in the video, there are a ton of clotting factors. So different clotting factors, there could be a mutation, they don't work properly, they don't get produced, that will produce the different types of hemophilia. In general, it's just a person is unable to form clots. It can be partially treated with injections of the missing proteins. We figured out how to do that. And it actually runs within the royal family. Queen Victoria was a carrier, and many, many, many of her children and grandchildren ended up with hemophilia. Blood typing is next. Blood types are determined by proteins on the surface of red blood cells. A blood transfusion is a transfer of blood from one person to another. We need to make sure that blood types are compatible to prevent a process called agglutination, which is the clumping of red blood cells. And we talked about before, red blood cells clump. They block important arteries and veins, tissues, certain tissues and organs. They don't get the oxygen they need, and that tissue dies, lead to a heart attack or a stroke, depending on where that blood vessel is. So our blood groups are called ABO blood groups as humans. First, we talk about what antigens are. An antigen is a foreign substance, often a glycoprotein that stimulates an immune response. All different pathogens have different antigens on their surface, and that is the specific piece that our body reacts to. Our body knows it's foreign, it's not supposed to be there. Blood types are determined by the presence or absence of two antigens, type A or type B. Type A blood has the A antigen. Type B has the B antigen, type AB has both, and then type O has neither. And at the bottom, it's just represented by different shapes or the absence of. It's just for visualization. Antibodies are protection proteins made by our B cells. They are specific and bind only to the antigen they are made for. So type A blood makes type B anti-B antibodies. People with type A blood, we do not want type B blood inside of our bodies. Same with all the rest of them. Type AB blood makes neither, and type O makes both. We have to know all this for compatibility purposes in case we ever need a blood transfusion or in case we ever need to give blood. So during a blood transfusion, if antibodies in the recipient's plasma bind to antigens on the surface of donated red blood cells, agglutination will occur. Therefore, type A cannot receive B or AB. Type B cannot receive type A or B. Type O can only receive other type Os, but can donate to everyone because they don't have any antigens. They don't have anything on the surface of their proteins. So they can donate to anyone. They're called the universal donor. And then type AB can actually receive any type blood. They have A, they have B, or type O doesn't have any, so it doesn't matter. So to be sure, a cross-match must be done where small amounts of blood are mixed together to test for agglutination. So these are blood compatibility charts just in general. O is the universal donor, can donate to everybody else. AB is the universal recipient, can receive from anybody else. And then on the right is a chart. When you see the little dots, that is agglutination represented. They're tiny little blood clots that are sticking together. The Rh factor is another antigen found on some blood cells. If it is present, we just say the blood is Rh positive. If it is not, it's negative. Unlike anti-A or anti-B antibodies, anti-Rh antibodies only develop if in a person after they are exposed to Rh factor, which doesn't happen very often except for childbirth. So when a woman who is Rh negative gives birth to a fetus that is Rh positive, because it is genetic, some Rh blood can leak from the fetus into the mother, will cause the mother to make those anti-Rh antibodies. Isn't a problem normally, but if they have a second child, and that second child is also Rh positive, the antibodies can cross the placenta and attack the fetus's red blood cells. This can lead to anemia and disabilities and even death in that fetus. So to prevent this, the mother receives a shot of a medication called Rogam, which is the anti-Rh antibodies, within 72 hours of giving birth to a child who is Rh positive. And then last but not least, the ever-present homeostasis. Many organ systems of the body interact with the cardiovascular system to maintain homeostasis. So the cardiovascular system and the lymphatic system, interstitial fluid originates from blood plasma. It's absorbed by the lymphatic capillaries, which becomes lymph. The lymph courses through the lymphatic vessels, eventually returning to the cardiovascular system. It works with the respiratory system for that gas exchange, the oxygen for carbon dioxide. It works with the digestive system, delivers nutrients from the digestive system to the interstitial fluid. It also removes metabolic wastes and delivers waste to our excretory organs, mainly our kidneys. Our muscular system works with the cardiovascular system because it contributes to blood movement. Cardiac muscle contractions circulate blood throughout the body. Smooth muscle contractions or relaxations and blood vessel wall changes in vessel diameter, helping maintain the correct blood pressure. Skeletal muscle contraction compresses cardiovascular and lymphatic vessels to make sure it's flowing through our bodies. Lymph returns to cardiovascular veins. Blood in the cardiovascular veins drains back to the heart. Works with the skeletal system. Red bone marrow produces the cells. Bones contribute calcium ions to the process of blood clotting. Works with the endocrine system because hormones course throughout our blood, and hormones regulate blood cell production, bone calcium release, and a host of other things. It works with the urinary system. The kidneys help regulate the acid, base, and water salt balances of the blood and interstitial fluid. And then that erythropoietin, the hormone produced by the kidneys, stimulates red blood cell production. And this is actually in your book. It's figure 6.12, but it is all the ways the cardiovascular system works with every single other system in the body to maintain homeostasis. That is all for chapter 6.